Having regulations in place for cell cycling is critical. When mutations cause cell cycle regulation to go haywire, cancer can be the result. And this is video 10.5 about cancer. We've got one big goal here, and that's to describe how normal cells can turn into cancer cells. But that story is a detailed one. From a big picture perspective, cancer is simply unregulated cell cycling. Cells divide uncontrollably, and that's what causes cancer. But what causes the uncontrolled cell division is mutations. Specifically, mutations in genes responsible for cell cycle regulation. To get a better sense of how cancer works, it's important to get some vocabulary straight. First is the big word tumor. A tumor isn't necessarily cancer. A tumor is just a ball of cells that ultimately came from one cell that was mutated. All of the cells in a tumor are daughter cells from the same mutated cell. Tumors can be benign, which means that the tumor stays in one place and doesn't threaten a person's health. Shown on the left here is a benign breast tumor. If a person has a benign tumor, this is not considered cancer. Often, benign tumors are just removed with surgery, and this is because benign tumors can change into cancer. But sometimes the tumor's just left there if it's not really causing a problem. Why risk the surgery? And just keep an eye on it. The other type of tumor is a malignant tumor. Malignancy is characterized by two properties: invasiveness and metastasis. The middle diagram shows how the tumor has turned invasive. Invasive simply means that the cells are breaking away from the tumor and spreading into nearby tissues. The diagram on the right shows how a tumor can turn metastatic. With metastasis, cells break away from the tumor, but what's different is that the broken away cells make their way into blood vessels or lymph vessels, which are over here. Once in the blood or lymphatic system, the cells can travel almost anywhere in the body. And then if those cells land in another tissue, then the cells can start new tumors in new places. A metastatic tumor can either be invasive or metastatic or both. If a person has metastatic tumors, then they're considered to have cancer. The types of genes involved in cancer mutations fall into two categories. The first category is called proto-oncogenes. Proto-oncogenes are normal versions of genes that have a job of stimulating the cell cycle. Growth factors are really good examples of proto-oncogenes that we've heard about. When a proto-oncogene gets mutated, however, now the proto-oncogene is called an oncogene. In the mutated form, the oncogene still stimulates cell cycling, but in an abnormal and accelerated way. The diagram at the bottom shows three ways that proto-oncogenes can malfunction and become oncogenes. First, if a mutation happens with the gene's promoter, the regulatory region next to a gene, the gene can be turned on at the wrong time, causing a cell to divide when it shouldn't be dividing. Second, if there's a gene duplication where multiple copies of the gene are put into the chromosome and different places are right next to each other, an oncogene can be turned on at too high of a level, causing a cell to divide more than it should. Finally, a mutation can cause the oncogene to change function. For example, imagine if a mutation caused a growth factor to irreversibly bind to a growth factor receptor. In that case, cell cycling would be turned on all the time, and that would be a big problem. An example of a proto-oncogene turning into an oncogene is called Wrasse. Wrasse is a relay molecule involved in growth factor signaling pathways. You can see the normal function of Wrasse up here. Here's growth factor ligand binding to a growth factor receptor, and hopefully you can see that this looks like a receptor tyrosine kinase, because upon ligand binding, the receptors dimerize and phosphorylate. But there's a downstream G protein called Wrasse. Once Wrasse is activated, the activated Wrasse activates a kinase cascade, and then that leads to transcription and translation of genes that promote the cell cycle and cause the cell to divide. But look at the bottom. Wrasse has now undergone a mutation, and it looks a little bit different. And Wrasse is active even though there's no growth factor ligand in the receptor, and the receptor hasn't been activated and phosphorylated. But the Wrasse mutated oncogene is still active. So even though the signal hasn't been sent for the cell to divide, the pathway to turn on cell division is still happening. While this diagram focuses on Wrasse, a mutation in any of these proteins in the kinase cascade or this receptor that causes the pathway to be artificially turned on can cause abnormal cell division. Another example of a proto-oncogene that could turn into an oncogene is a cyclin. Remember from our last video that a cyclin normally causes cell division only when the cyclin levels are really high. For example, when this MPFcyclin is at its highest level, the MPFcyclin binds to the MPFCDK and activates the MPFCDK, allowing the cell to pass through the G2M checkpoint. Imagine, though, if there was a mutation that caused MPFcyclin to be around all the time. Now, MPFcyclin would be considered an oncogene. Activate CDK all the time, and the cell could pass through the checkpoint even if it's not supposed to. Besides proto-oncogenes turning into oncogenes, the other type of gene class that is involved in cancer is called tumor suppressor genes. Tumor suppressor genes normally work by stopping a cell from dividing, inhibiting the cell cycle. For example, P53 stops a cell from dividing when DNA is damaged. When mutated, however, tumor suppressor genes no longer stop the cell from dividing. This means that the mutation in a tumor suppressor gene stimulates cell division by absence of suppression. Perhaps the most famous example of a tumor suppressor gene is P53. Normally, P53 is activated by DNA damage. When the DNA is damaged, P53 stops the cell cycling and activates DNA repair enzymes to repair the DNA. If the DNA is repaired, the cell cycle is allowed to resume and carry on as normal. But if the DNA isn't repaired, P53 then activates a separate set of proteins that kill the cell by a process called apoptosis. In the bottom, though, P53 is mutated. Then the cell can divide even if there's DNA damage. Hopefully you can imagine how dangerous this would be, because a mutation in P53 makes it more likely that other mutations would happen and really increases the chance for cancer. What's fascinating is that over 50% of cancerous tumors have P53 mutations in them. So why is cancer so scary and bad in the first place? Well, first of all, there's so many different types of cancers. This diagram shows a bunch of different types of cancer, but that's just the tip of the iceberg. Moreover, for example, each type of lung cancer isn't the same as the other. Different lung cancers can be caused by different mutations and different genes, but all would be classified as lung cancer just based on the fact that the tumors happen to be found in the lungs. Another reason why cancer is bad is because tumors can steal nutrients from healthy cells, as shown down here. This diagram shows a tumor in purple that starts to secrete angiogenic factors. Next, blood vessels start to branch out to feed the tumor. Not only do they feed the tumor, but they surround the tumor. The process of new blood vessel formation is called angiogenesis. A tumor getting its own blood supply is very dangerous for many reasons. First of all, the blood supply allows the tumor to grow even faster, with a mainline stream of nutrients. Second, having blood vessels close to the tumor will make it easier for the tumor to metastasize. Third, it'll be a lot harder to surgically remove the tumor if there are large blood vessels wrapped all around the tumor. So angiogenesis can mean bad news. Finally, cancer is bad because having a tumor someplace can interfere with normal cells that are neighbors to the tumor. A really clear example, I think, is a brain tumor. In this diagram, you can see a whole bunch of different types of brain tumors, all in green. And all of those tumors are neighboring normal brain tissue. Since a skull only has so much space, the growing tumors can push portions of the brain against the hard skull and cause symptoms like blurry vision, dizziness, nausea, headaches, and other weird symptoms. With all this talk about cancer, it's easy to get really freaked out. So now I want to tell you why not to get freaked out. Cancer doesn't happen in all just one step with a snap of the fingers. Cancer takes many steps, many mutations. Moreover, all of those mutations need to land in the same exact cell, and those mutations have to land in certain genes that are involved in cell cycling. Next, even if there are mutations, cells have mechanisms to repair the damaged DNA. Finally, even if that fails and the mutations escape repair, most of the time cells die by apoptosis rather than keep on dividing and accumulating more and more mutations. So there's a lot of safeguards in place. Let's look at an example of this multi-step process. Here in this diagram, we see a sheet of epithelial cells. Let's imagine these cells are in your stomach lining. You see that just one of the cells gets a mutation, and it makes this look like an orange cell. Maybe this turns a proto-oncogene into an oncogene, or it's a mutation in a tumor suppressor gene. You can see that the orange cell has divided and made a bunch of orange daughter cells. The mutation hasn't spread. That mutated cell has just made more cells. But now, one of the orange mutated cells gets a new mutation, as marked by the green cell. The green cell is now dividing a lot faster. It didn't kill the orange cells, but green cells are dividing so fast that the orange cells are kind of inconsequential now. All of the daughter cells of the green cell also have the green mutation. Another thing that's happened is that the green cells are now growing in a pile. This means that the green cells have lost anchorage dependence, and even though they're not attached here, they're still dividing. We would officially call this cell pile a tumor. Since the tumor is in the same place where the first mutated cell started, this is called a primary tumor. And if you look here, you can see that one of the green cells now has the purple mutation. This purple mutation is really bad news. This diagram makes it seem like all of the green cells turned purple, but that's not the case. Think about it as the purple cells dividing so fast that the number of green cells are now pretty insignificant. Anyway, the purple cells now have gained the ability to invade nearby tissue. This tumor is invasive. Not only that, but purple cells have broken off of the main tumor and gained access to nearby blood vessels. This tumor is metastasizing. Since the tumor shows invasiveness and metastasis, now this tumor is malignant, and this is officially cancer. This final diagram shows the fate of the tumor cells that made it into blood vessels. The tumor cells exit the blood vessel in a new place and have made a tumor in this new place. Since this tumor is in a different place away from the original tumor, this new tumor is called a secondary tumor. Knowing what you know about cell cycle and cell cycle regulation, imagine all the different mutations that can give cells the ability to divide faster. Mutations that code for or that cause cyclins to be turned on, DNA replication to be turned on, or angiogenesis to be turned on can all help cells divide faster. Mutations that cause p53, apoptosis genes, or contact inhibition genes to be turned off can eliminate shutdown procedures that would stop cancer. But it takes accumulation of mutations in those genes and other genes to cause the cancer. It's currently estimated that a cell needs anywhere between about 6 and 80 mutations to become a cancer cell and a cancer tumor. Moreover, at least one of the mutations needs to be in a proto-oncogene to make that an oncogene, and one or more of the mutations has to be in tumor suppressor genes. and mutations accumulate over time. And this is why most cancers are more common in older people than younger people. The longer you live, the more mutations you get. The more mutations you get, the more likely you are to have cancer. Remember, though, that cells that get mutations can often fix those mutations, or, if not, often die by apoptosis. Apoptosis is a process where a cell kills itself. The word apoptosis comes from a Greek word meaning falling off. Scientists think of cells undergoing apoptosis as kind of like leaves falling off of a tree, where it's part of the normal life cycle. Apoptosis isn't just a way to kill cancer cells, but also happens to cells that are infected or damaged, or cells that just aren't needed anymore. Other common ways to describe apoptosis are programmed cell death or cell suicide. You can see the process of apoptosis play out in the diagram at the bottom. A normal cell can undergo apoptosis, and then physical changes start happening. The DNA is chopped up, and the cell starts to become deformed, a process called blebbing. Finally, the cell breaks apart into apoptotic bodies, and those pieces are found and digested by macrophages to clean up the mess. There are lots of causes for DNA mutations. One common cause is simply errors that happen when the DNA is copied during S phase of interphase. Errors caused by DNA copying in cell division are called spontaneous mutations. There are a lot of environmental factors that cause mutations as well, and these are called carcinogens. You've probably heard about things like UV rays in the sun, or X-rays, or smoking cause cancer. And some viruses actually contribute to cancer as well, like the link between the HPV virus and cervical cancer. If a mutation is found in a sperm or an egg, then the mutation can be passed from parent to offspring or inherited. This means that the offspring will be predisposed or have a higher likelihood of the cancer, as there are now one less step in the multi-step pathway leading to cancer. Besides surgery to remove tumors, two of the most common cancer treatments include radiation therapy and chemotherapy. Radiation therapy uses beams of radiation directed right at a tumor. The goal is to put even more mutations in the cancer cells so the cancer cells will die. To use radiation, doctors have to know the exact location of the tumor so they know where to blast the radiation. Radiation therapy is often coupled with chemotherapy. Chemotherapy puts chemicals into the bloodstream that are toxic and target rapidly dividing cells. There are a lot of different chemotherapy strategies, but all of them are directed against different steps of mitosis. For example, some chemotherapy drugs are fake DNA bases. If a dividing cell puts a fake DNA base in, the DNA will no longer function in that cell. Other drugs target the mitotic spindle, either to stop the spindle from forming in the first place or keep the spindle from breaking down. If the spindle is stuck in its fully formed state, then a dividing cell will never get past metaphase. So let's recap our whirlwind tour through cancer biology. Cancer is a multi-step process that involves mutations that increase the rate of cell division. Put simply, cancer is out-of-control mitosis. Normal versions of genes that cause cells to divide are called proto-oncogenes, but when proto-oncogenes mutate, they're called oncogenes. Oncogenes speed up cell division, but in an abnormal way. Tumor suppressor genes normally keep the cell from dividing too fast. When tumor suppressor genes are mutated, though, cells can skip critical checkpoints and pile up more mutations. We learned about the important process of apoptosis, a self-destruct mechanism to kill cells that are damaged or not needed anymore. Finally, we learned how medicine has taken advantage of knowing about the cell cycle and how it works to disrupt cell division in cancer cells.